So when we're talking about growth of microbial populations, now unlike humans or dogs or most animals, when we talk about growth in a human, we're talking about baby to adulthood. So we're talking about change in size and stuff like and things like that. Change in development, that kind of thing. But when we're talking about microbial growth, what we're usually talking about is replication. There is a slight change in size, but when we're talking about growth of microbial populations, we're usually talking about growth in numbers of microorganisms. All right, so that's kind of a little different. So when we talk about growth and we talk about replication, the way a bacteria cell replicates is called binary fission. So let's go through the steps. Okay, so I have a bacteria cell, cytoplasmic membrane, cell wall, and then the DNA, nucleic acids in there. Now we know there's not a nucleus, right? No nucleus in prokaryotic cells, but there is a nucleoid, a nuclear region where the DNA is hanging out. It's all part of the cytoplasm. So in order to replicate, the first thing that has to happen is we have to copy those instructions. We have to copy the DNA. So the chromosome replicates. We do see a little bit of enlargement of this in preparation for replication. So we've got two chromosomes now. They move to the opposite sides of the cell and kind of take a hold of the cell membrane to keep on the opposite sides of the cell. The cell membrane and cell wall invaginates but forms this septum formation as it comes in, and then as that completes, we then have two completed cells. This is called binary fission. Now it's going to look very similar to another process. What process does it look like that you already know? Kind of looks like mitosis. Mitosis, right? DNA replicates, it divides, blah, blah, blah. Mitosis is the name that describes the nucleus replicating in a eukaryotic cell. So that is not the same thing as binary fission, but the steps are very similar. But do not get confused and call this mitosis. This is not a nucleus, so therefore it is not mitosis. The proper name for this is binary fission for that. All right, so the time it takes to go from one cell to two cells, okay, those are just the things. DNA replicates, attaches to walls, elongates, builds that septum dividing wall, splits into two cells. This is not mitosis. So the time it takes to do this is one cycle. The time it takes to go from one cell to two cells is that time. If I add, this is called the generation time. I think I've got a slide for that. Let's keep going. All right, so binary fission, we understand that. When we're talking about bacteria cells replicating, we go from one cell to two cells, and then both of those two cells undergo binary fission, and so now you have four cells. All four of those cells undergo binary fission and replicate, and now you have 8 cells. This is called a logarithmic growth scale. So you can contrast this with, keep pushing the wrong buttons. You can contrast this with arithmetic growth. This is typically what you see in larger mammals. You have one, and then they produce one more, and you get another one, so you get 2, and then you add one more, and that gets 3. This is a very slower growth rate, but this logarithmic growth rate can get into scary numbers very quickly. So when we're talking about bacterial growth, and we're talking about logarithmic growth, we get into scary numbers very quickly. And I will go back to that last slide. You can see we started with 1 in 30 minutes, it replicated. So now we have 2 cells after 30 minutes. After 60 minutes, 30 minutes plus another 30 minutes, we went from 2 cells to 4 cells, 4 cells to 8 cells, 8 cells to 16 cells. You can see that growth happens very quickly when we're talking about logarithmic growth. All right, so when we're talking about us populating the planet versus bacteria, their ability to replicate is really kind of scary. So why, if bacteria can replicate logarithmically and they can keep, every time they replicate, they double in number, why aren't bacteria taking over the world? Why aren't we actually knee-deep in bacteria? We see colonies forming on a plate. Why aren't bacteria just taking over everything? And the reasons why is that they start competing for nutrients. They can replicate in a logarithmic fashion as long as there's enough nutrients to support the population. So that's what's going on. Once you get a number, more bacteria, then there are nutrients to support that bacteria, that bacterial population, then you start getting cell death and you don't end up with this logarithmic growth forever. So that's good. It is growth limiting. All right, so let's do the vocabulary that I started to introduce earlier. The time it takes to go from one cell to two cells, this is called your generation time. So it's the time it takes for a cell to divide, or you can also think of it as its population is doubling. This is called the generation time. So in that original slide that we were looking at, 30 minutes was the generation time for that particular bacteria. Now this can vary between different bacteria. E. coli has a very quick generation time. It can replicate as fast as every 20 minutes. Tuberculosis and leprosy, they can take up to 24 hours to replicate. They're very slow growing. We introduced that with that Mycobacterium tuberculosis at the introduction of chapter 3, and we talked about that it's very slow growing, and drugs only work while they were growing. And so it takes a very long series, 6 months to up to a year of drugs to get rid of all that Mycobacterium tuberculosis. So again, a lot of different variations in the generation time, depends on the species. It also depends on the growing conditions. When conditions are doing well and there's lots of nutrients, that generation time can happen a little bit more quickly. When nutrients are scarce, or maybe temperature differences. Remember we talked about metabolism slowing down, and so if temperature is not right, pH is not right, that's going to slow down their metabolism and their rate of growth, and so that is going to influence the generation time for a species. Okay, so genetics, species to species, is going to influence this, but also environment plays a role in the generation time. All right, so let's talk about phases of microbial growth and define those. We've already talked about this logarithmic growth phase. When nutrients are plentiful and all conditions are good, you get that logarithmic growth. You get that doubling in numbers for every generation time, and you can see this exponential growth phase. So this is considered the log phase of bacterial growth. We're looking at growth over time. We're looking at the number of cells, and in this case, it looks at living cells for that. In your labs this week, you're going to talk about spectrophotometry, and that works on absorption, and that doesn't can't tell the difference between a live cell and a dead cell. But in this one, we're talking about this growth phase. We're looking at only living cells from that. So this is the number of living cells. So logarithmic phase, you can see that number is increasing exponentially because conditions are favorable. Now, at some point, the population is going to reach saturation. You're going to have only so many limited nutrients. We talked about nitrogen being a limiting nutrient. We talked about, when we talked about that lactobacillus in the female vaginal region, we talked about estrogen being a limited nutrient. When that happens, then the cells are going to start competing for that. Some are going to win and get that nutrient, and some are going to lose. And when they lose, they're going to die. So this is what's happening during the stationary phase. Now, it's not that replication is not occurring, because when you see a flat line, it's tempting to think, oh, they're not replicating, and that's not true. Replication is still occurring. What's happening is the rate of replication equals the rate of death. So for every one cell that gets the nutrients and is able to divide into two cells, there's another cell that didn't get the nutrients and dies. So you went from two cells, one that won the nutrient, one that didn't win the nutrient. The one that wins the nutrient, here, let me go back to the live. So I have two cells. One is getting the nutrient. It is able to replicate and becomes two cells. This guy lost, didn't get the nutrient, ends up dying, and my net result is still two cells. That's a stationary phase, and you see that kind of level off, and you get that. So the population is stationary. The death rate equals the replication rate in that phase. All right, so that will happen for a while, as long as nutrients keep coming in to support that. But what happens if you're in a closed system, say I'm in a test tube, eventually all the nutrients are going to be used up, and more cells are going to lose the competition. More and more cells are not going to be able to get the nutrients that they're going to capable to live and they're going to die. Then we start declining in numbers. And if I go back to my fingers, this time let's give me 3 cells. I got 3 cells, wish I had another hand. Got 3 cells, and this one doesn't get enough nutrients, he dies. This one doesn't get enough nutrient, he dies. This one does, and he replicates, but now I have 2. And you can see how my number of cells is declining as that occurs. So nutrients are limited, and we are now in the death phase. Again, it's not that replication isn't still occurring, it's just the amount of death is greater than the numbers produced by replication. So this is the death and decline phase in bacterial populations. All right, so I skipped this first step. Let's go back to this one. Before we get logarithmic growth, what we see in a population is something that is referred to as a lag phase, and you'll notice it's also a stationary phase. Now, the lag phase is different than the stationary phase. Remember, the stationary phase I defined as the replication rate equaling the death rate. The lag phase is something different that bacterial cells undergo and what we observe. All right, so say I take a cell here. Before I do that, let me relate it to something that's easier to do. So I am going to move. I live here in Cyprus. I've decided to move. So I'm going to move to Michigan or someplace, really cold. And that is a big move. There's a lot of things. You can even take it in another country if you want to. There's going to be a lot of things that I need to do as I'm moving. To prepare for that. I'm going to need to buy some new clothes. I don't have cold weather clothes. I'm going to have to find new doctors. There's going to be new grocery stores. There's going to be different types of grocery stores. H-E-B is not up there. What am I going to do? I'm going to have a lot of things that I have to figure out in order to grow and thrive in my new community for that. So this is what's going on in a lag phase. I take a bacteria cell and I inoculate it into a new culture. And it's going to see what nutrients are available in its new neighborhood. And that's going to take a little bit of time. And at that point in time, replication actually doesn't occur. The bacteria cell is kind of seeing what kind of nutrients are there. It may need to make some new plans, buy some new clothes. It may need to produce some new enzymes. It comes from a neighborhood where there was a lot of glucose. It moved into a neighborhood where there was very little glucose and a lot of lactose. Well, it can handle that. It's got a plan B. It can utilize lactose, but it's going to need to make some new enzymes, right? Plan B, we talked about that with the metabolic pathways. It's going to need to make some new enzymes, and that's going to take some time, right? I got to transcribe them and then translate them into proteins and then get them out there and do their job. It's going to take some time. So replication doesn't occur in that phase. This is defined as the lag phase for bacteria. So it's a stationary phase. Replication is not occurring, and the cell is using it to identify what nutrients are in its environment and produce any enzymes that it may need to be successful in its new environment. So how long does this lag phase last? Well, if I move to Michigan, there's going to be a lot of adjustments that I'm going to have to make. If I move from one side of 290 to the other side of 290, stay in Cyprus, there's very little. I can still go to H-E-B, I can still go to my same doctor, there's going to be some adjustments, but not much. So the lag phase, if the environments are very similar, the lag phase can be a very short amount of time. If the environments are very different, then that can extend the lag phase for every bacteria as it adjusts to an environment. So that's what determines how long the lag phase is. If it goes again from a very similar environment, maybe a very short lag phase if there.